Hello, my students. I'm your biology teacher, Chef Govind. Our topic today is Chapter 9, Ecosystem, and our subtopic is 9.2, Populations of Ecology. Here are the learning standards. Learning standard number 1, describe factors affecting population distributions. And number 2, analyze data to estimate population size of organisms using number 1, Quadrat sampling techniques, and number 2, capture, mark, release, recapture techniques. Population ecology. Population ecology is a branch of ecology which study the interactions of a populations with its surrounding. In previous lesson, in subtopic 9.1, we learned about population. Population is a group of organisms of the same species living in the same habitat. Population distribution show how the organisms of the same species are able to spread out in a habitat. Okay, factor affecting population distributions. Population distribution is affected by abiotic factor, which are, we can divide it into two, the effects of abiotic factor toward the plants and the effects of abiotic factor toward animals. So the effects of abiotic toward plants can be divided into 5: temperature, pH of soil, water, light, and mineral salt content. Whereas the effects of abiotic factor toward animal, only 4 factor, which is temperature, breeding site, water, and food supply. Factor affecting population distributions of plants. Okay, we look factor number 1, temperature, and the effects towards plants. The optimum temperature for plant growth is between 25 degrees Celsius to 30 degrees Celsius. Okay, beyond the optimum temperature, any increase in temperature will no longer increase the rate of reactions. High temperature can cause enzymes to denature. Biochemical processes in plants are disrupted and there stunts the growth of plants, increase the rate of water evaporation by transpiration, and slow down the rate of photosynthesis. Low temperature reduces the activity of enzyme, which slow down the biochemical reactions. At low temperature, an enzyme catalyzed reactions take place slowly. Okay, we look at factor number 2, water. Water is needed for enzyme activity, photosynthesis, and transport, and also support in herbaceous plants. Dry regions such as desert and tundra area have low populations distribution and also low density of plants. Light. Light is very important for the process of photosynthesis in plants. Areas which receive little light have a lower number of plants. This diagram shows how light affects plants' growth. The best wavelength of visible light for photosynthesis is blue and reds. Factor number 4, mineral salt content. Salt content affects the absorption of water through osmosis by the roots. High salt content in soil will cause plants to lose water through osmosis. Minerals are needed for the production of proteins, enzymes, nucleotides, vitamins, and other compounds. This diagram shows how different types of minerals affects the plant's growth. pH of soil. The pH of soil is important for nutrient absorption by the roots. Soil in which the pH is either too acidic or too alkaline will cause lower absorption of nutrient by plants. The growth of plant will be disrupted due to lack of nutrient. Look at this diagram. This diagram shows the proper pH level for growing grass. So this is the ideal for lawns, which is slightly acidic. The reading is 6.2 to 7. Factor affecting population distributions of animal. Factor number 1 is temperature and effects toward animals. Areas which are exposed to high temperature in habitat are less inhabited by certain animals, such as worms and snails. Factor number 2, water. Animals are concentrated in areas that have enough water sources for drinking and cooling the body. Okay, this one. Breeding site. A safe and suitable breeding site is needed by animals to raise their offspring. And last one, factor number 4, food supply. Food is important for survival because animals are heterotroph, which depends on plants and other animal foods. There are 3 patterns of population distributions, which are clumps, randoms, and uniforms. Clump organisms form separated groups, okay, like these diagrams. This condition is caused by uneven distributions of resources in an area, for example, water and foods. Randoms. Organisms are distributed in a free pattern in area. Distributions involve species that have little interactions among individuals. Okay, this is diagrams. Okay, randoms. Okay, uniforms. Organisms are distributed all over inhabited area. This distribution involves species in which every individual compete for limited resources in an area. Okay, this one. Okay, so look uniforms in this area. Estimations of organism population size. Two important factors for studying population ecology are population size and population density. What is population size? Population size is the number of organisms present in the populations, and population density is the number of individuals of a species per unit area of a habitat. Okay, the population size in area can be estimated by a random sampling techniques when the study area is too large and difficult to obtain data. A sample which represents the area is taken to give an estimation of the abundance and the distribution pattern of certain organisms that inhabits the area. There are two sampling techniques we learn in these lessons: Quadrat sampling techniques and capture, mark, release, recapture techniques. Okay, what is Quadrat sampling technique? Quadrat sampling techniques is technique can estimate the population size of land plants or animals which are inactive or move slowly. The number of organisms in the quadrat area is the sample which represents the whole area of study. The data obtained from the quadrat can be used to estimate the populations in the area of study. While capture, mark, release, and recapture techniques can estimate population size of organisms such as foxes, sharks, snails, millipedes, insects, butterfly, which can move freely in the studied area. Quadrat sampling techniques. A quadrat is a square frame structure like this one. This is made of woods, iron, or plastics. Normally, a quadrat is subdivided into few parts of the same size to estimate the coverage percentage of any species. Okay, the size of the quadrat depends on the types and size of organisms, the area of study, as well as the distribution and density of the plants being studied. For example, this is 1 meter square quadrat. Is suitable to estimate the populations of plant in the school field. This one is 50 centimeters square. This one is suitable for small area. Okay. During a quadrat sampling, you should place a quadrat randomly and not based on an area with the most plant being studied can be found. Okay, you must put the quadrat randomly, and okay, you cannot choose the place. Okay. Okay. Quadrat sampling techniques. Quadrat sampling technique can estimate the frequency, density, and coverage of any plant species which is being studied in the area of study. So this is the formula of frequency, this is formula for density, and this is coverage percentage. Okay, we look one by one how to use this formula in our activity. Activity 9.3. The aim of this activity is to carry out a field study to estimate the population size of plants in a school fields. Okay, the apparatus, a quadrat measuring 1 meter times 1 meter, a pen, a notebooks. The procedure, number 1, identify a plant species X present in the school fields. A quadrat measuring 1 meter times 1 meter is randomly placed in a part of the school fields. Calculates the number of species X in the quadrat. Repeat step 2 and 3 at 9 other sites of the fields, randomly selected also for the same plant species. Record the result in the table. And number 6, calculate the frequency, density, and the coverage percentage of the sample species X. Okay, this is the tables. Okay, there are 10 quadrats. Okay, the total quadrat containing of species X, and we need to know the frequency of species X. Okay, this is the formula. Okay, frequency, the number of quadrat containing studied species, and the total number of quadrat used times 100%. Okay, this is example of result we obtained. Okay, frequency is the probability to get n individuals of any plant species in every quadrat. Okay, in quadrat number 1, there is a species there. Okay, the species X is present. Quadrant number 2, species X is present. Quadrant number 3, presents. Number 4, presents. Number 5, species X also presents. Number 6, presents. Number 7, yes, presents. Number 8, species X is present. Number 9, yes, and number 10 also presents. So all quadrat presents species X. Okay, based on the result, okay, the number of quadrat containing studied species, there are 10 quadrat, all contain the species X, and the total number of quadrat used is 10, so time 100%. So the answer is frequency of species X is 100%. Okay, now we calculate the density of species X. Okay, this is the table. Okay, and this is the formula for density. Total number of individual species studied in all quadrat divided into total number of quadrat used and time area of the quadrats. Okay, so this is the result of the experiments. Okay, in quadrat 1 there are 3 species X. Okay, we put here the 3, and in quadrat number 2 only 1, quadrat number 3 only 4, quadrant number 4, 2 species X. Quadrant number 5, there are 4 quadrat X. And then quadrant number 6, there are 1 only here. Okay, we put 1 here. And quadrant number 7, there are 5. Okay. Quadrant number 8, 4 only. Quadrant number quadrant number 9, 3. Quadrant number 10, 3. So altogether is 30 species of species X. Okay. So how to do it? Okay, so the formula, total number of species X is 30. Okay, total number of individual species studied in all quadrat is 30. And then the total number of quadrat used is 10. Okay, and then area of quadrat is 1 meter square. So the answer is 3 individual per meter square. Okay, this is the answer. Okay, now we calculate the coverage percentage of sample species X. Okay, this is the formula for coverage percentage. Area covered by studied species in all quadrat divided into total number of quadrat used times area of quadrat times 100%. Okay. Coverage percentage is the percentage of soil surface covered by the plants. Okay, this is 1 meter square. Okay, there are 10 small square here, and also here are 10 small square. So altogether is 100 small square here. So this quadrant number 1, 3 only plants X occupy over hundreds small square here. So we put 0.3. Okay, quadrant number 2 only 1. Okay, we put 0.1. Okay, quadrant number 3 there are 4 species X over hundreds small square, so we put it as 0.4. Quadrant number 4 only 2, okay, so we put 0.2. Okay, number 5 is 4, we put 0.4. Number 6 is 1 only, we put 0.1. This one is 0.5 because 5 species X in quadrant number 7. Okay, quadrant number 8 is 4, 0.4. Quadrant number 9, 0.3. And quadrant number 10 is 0.3. So altogether is 3. Okay. Okay, the calculations. Okay, coverage percentage of species X in school fields. Area covered by studied species in all quadrat is 3. Okay, total number of quadrat used is 10. And area of quadrat is 1 meter square, so time 100%, so the answer is 30%. So the coverage percentage of species X in school field is 30%. Discussion question. Question number 1, state the pattern of species distributions that you have studied and justify your answer. So based on what we study, This is random distributions. Organisms are in a free patterns in an area. Question number 2. State the surrounding factor which affect the patterns of population distributions of the species that you have studied. The answer is temperature, light intensity, and humidity. Question number 3. What is the frequency, density, and coverage percentage of the species in the school field? The frequency is 100%, density is 3 individuals per meter, and coverage percentage is 30%. Captured, marked, release, recaptured techniques can estimate the population size of organisms such as foxes, shark, snail, millipede, insect, and butterfly, which can move freely in the study area. This technique involves a number of steps such as animals under study are captured randomly in the area of study, masked in natural habitat. The number of animals captured is recorded, and then every animal captured is marked by using nail polish, paint, or Indian inks. The marked animals are then released into the same area of study. After a certain period of time, a second capture is done randomly in the same area of study, and then the total number of animals captured the second times, both marked and unmarked, is recorded. So this is how we mark the animal in our study area. Okay. Activity 9.4. The aim of this activity is to carry out a field study to estimate the population size of animals. The apparatus: paintbrush, Indian ink or nail polish, a pen and paper. The procedure: choose a suitable area in the school compound where you can carry out a study. Identify the type of animal which is abundant in that area to become a sample. Capture 5 samples of the chosen organisms in the study area. Mark all the captured organisms. Release all the marked organisms in the same area where they have been captured. After a few days, recapture another 5 samples of organisms in the same area. Record all the marked samples in the second capture. Estimate the population size of the organisms by using the formula. This is the formula: population size, P times Q divided by R. P is the number of animals in the first capture, and Q is the number of animals in the second capture, and R is the number of marked animals in the second capture. Okay, so this is the formula. This is example of table that we can use to record the data. Let's say we study the population of centipede in science gardens. In first capture, we get 36 number of centipede in the first capture. In the second capture, there are 40 centipedes, but the number of marked centipede in the second capture is 12. So to calculate the population size is 36 times 40 divided by 12. So the answer is 120. So the population size of centipede in science garden is 120. Okay, we must use this formula. Okay, so discussion question: what is the substance that you use to mark the captured animals? Justify the substance that you have chosen. Okay, the answer is nail polish, because nail polish does not harm the organisms and cause death, and nail polish also not cause migrations to another area. Okay. Question number 2. State the interactions that have happened between the study organism with biotic and abiotic component in that area. Biotic feed on smaller insects. Small plants take shelter from sunlight. Abiotic: oxygen to breathe. Water maintain the humidity of skins. Stone for hiding. Question number 3. State the basic need that enable the studied organisms to inhabit the area: oxygen, food, and water. Activity 9.5. Pleurococcus is a type of green algae growing on areas that are protected from the scorching sunlight to form a slimy layer on places such as tree barks, stone, and moist soil. This algae can reproduce very fast through vegetative cell division by binary fissions. Pleurococcus is a sphere-shaped unicellular organism which can exist individually or in a group. It has thick cell wall to prevent excessive water loss. Every cell is abundant with chloroplasts for photosynthesis. The aim of this activity is to carry out a field study to observe the effect of abiotic components on the populations of organisms. The apparatus, 5 Quadrat 10 cm x 10 cm, which are made from transparent plastic, a marker pen, a roll of string, and a meter ruler. The procedure: choose a tree which has an uneven growth of Pleurococcus at the lower part of the tree trunks. Identify the site of tree which receives the most sunlight. Tie a string around the tree trunk at an estimated distance of 1 meter from the ground. Draw grid measuring 1 cm x 1 cm on each of the transparent quadrat. Put the quadrat at the lower parts of the strings at different aspects P, Q, R, S, and T, like this. Okay, we tie it. Okay, and 1 meter from the grounds. Okay. Procedure number 6. By using a marker pen, shade all parts where the growth of Pleurococcus can be seen in the transparent quadrat. Number 7. Count the number of squares which contain Pleurococcus in the quadrat. You must take note, only count squares which have half cover or more than half cover. Okay, then we can count it. Less than half, we not count it. Okay, number 8. The cover percentage for all quadrat is estimated by using the formula. So this is the formula: coverage percentage, area covered by Pleurococcus in 5 quadrat. Okay, we use 5 quadrat here. And then total number of quadrat used is 5. Area of quadrat is 100 centimeter square because we use size of 10 by 10 centimeter. Okay, and then this is the tables that we can use to record all the data. Okay, so this is the result. Okay, this is the result. Okay, this is the sample of actual answer. Okay, quadrat P. This is quadrat P, Q, R, S, and T. Okay, I will show you how to count the total coverage of Pleurococcus based on this sample. Okay, let's see how. Okay, number 1, 2, 3. Okay, this one we cannot count it because not more than half square. Okay, so this one 4, 5, 6, 7, 8. Okay, this one not more than half. 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46, 47, 48, 49, 50, 51. Okay, 51's total coverage of Pleurococcus in quadrat P. Okay, the Q is 78. Okay, R is 37, S is 70, and T is 47. Okay, we try to count R. Okay, because R is the last one. Okay, 1, 2, 3, 4, 5, 6, 7. Okay, this one cannot. 7, this one okay. 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37. So 37, correct. Okay, so you can count S and T by yourself. Okay, so the total coverage of Pleurococcus in all quadrat is 283. So this is the calculation. 283, okay, divided by 5. 5 is the number of quadrat, and 100 centimeters square is the size of the quadrats, and then time with 100%. So the answer is 56.6% coverage percentage of Rhococcus in 5 quadrats. Okay. Discussion question. Question number 1. Which quadrat receives sunlight? A, the most, B, the least. Okay, based on this diagram, you can see this is the sunlight. Okay, P is directly exposed to sunlight, and then Q is 78. Q is the highest total coverage of Rhococcus. Okay, Q, and then we can see R is at the middle of tree trunk. R is the lowest total coverage of Rhococcus, which is 37 only. Okay, and S is the second highest, 70, and T is 47. Okay, based on the quadrat received sunlight, the most is P, and the least is R, because R is at the middle of the tree trunk. Okay. Question number 2. Which quadrat has coverage percentage of Rhococcus that is the highest? The highest is Q, okay, and the lowest is R, okay, because R receives less sunlight, okay, but Q is at the middle of P and R, so the temperature and the sunlight is suitable for the growth of Pherococcus. So Q is the most suitable place for the growth of Pherococcus. And then question number 3, explain the influence of sunlight toward the growth of Rhococcus. The population distribution of Rhococcus is higher in the area of moderate light and temperature distribution because rate of photosynthesis is the highest. When the light intensity and temperature are high, the outer layer of tree trunk becomes hot and dehydration of Rhococcus cells happens. The growth rate of Rhococcus becomes slower. Okay. Formative practice 9.2. Question number 1. An ecosystem has a population size, population density, and population distribution. A. What is the difference between population size and population distribution? Okay, the answer is population size is the number of organisms present in the population, whereas population distribution is the number of individuals of a species per unit area of habitat. Okay. B. State the factor that can affect population distribution. Temperature, water, pH of soil, salt content, mineral contents, reproduction place, and food supply. Question C. State the patterns of the population distribution and explain it. Clump for organisms forming separate group because of uneven distribution of resources. Randoms, organisms are in a free patterns in an area due to lacks of interaction with each other. Uniform, organisms scatter all over the area because every individual compete for limited resources in an area. Okay, question number 2. Quadrat sampling can be used to find the coverage percentage of plants in the school field. You are supplied with a quadrat which consists of a square-shaped wood frames. According to your biology teacher, the quadrat need to be modified to obtain a more accurate estimation. Explain. Divide the quadrat into smaller square parts of the same size using wire or string. Small squares which are covered by species either half or more are counted as having coverage, whereas lesser than half are ignored. That's all for today. See you in the next lessons. Thank you and bye-bye.